Hello everyone, welcome to this tutorial on phylogenetic tree analysis. Today, I'll guide you through our pipeline submodules, step by step, to run code and interpret results. By the end, you'll understand the full workflow. Let's get started. On the left, you'll see our 4 submodule files. Let's begin with Submodule 1. This submodule introduces phylogenetic trees visualizing evolutionary relationships and their role in biology. We'll run Python scripts to explore tree structures. Key concepts. What are phylogenetic trees? They show how species, like primates, are related. Why are they important? They reveal evolutionary history, species divergence, and virus spread. How are they created? Using genetic or phenotypic data. Newick format, a standard for representing trees. Tree types: rooted, unrooted, cladograms, phylograms, and dendrograms, each with unique applications. Let's run code to see these in action. Code cell 1, rooted tree, COVID-19 variants. Let's run this cell to display a rooted tree of COVID-19 variants. I'll go to the Run menu and click Run selected cell. Here's the output. It shows variants like alpha, delta, and omicron, with branch lengths indicating evolutionary distances. This helps track virus evolution. Code cell 2, rooted tree, mammals. Next, a rooted tree for mammals. I'll press Shift plus Enter simultaneously to run the selected cell. This tree shows relationships among humans, chimpanzees, dogs, and more. Humans and chimps share a closer ancestor, while dogs diverge earlier. Code cell 3, exercise solution, avian species. Let's run this cell to load an exercise solution. Here's a rooted tree for birds like sparrows and eagles, great for practicing tree interpretation. Code cell 4, unrooted tree, COVID-19 variants. Unrooted trees show relationships without a common ancestor. Let's run this cell to visualize genetic similarity among COVID-19 strains. Here's the result, highlighting variant connections. Code cell 5, complex unrooted tree. Now, a more complex unrooted tree with primates, canines, and birds. Let's run this cell. This groups species like humans, dogs, and eagles, showing their relationships. Code cell 6, cladogram, primates. Cladograms focus on branching order. Let's run this cell to see primate relationships. This shows humans and chimps as closest relatives, then gorillas and orangutans. Code cell 7, cladogram, vertebrates. Another cladogram, for vertebrates. Let's run this cell. It groups frogs, lizards, birds, mammals, and fish by branching patterns. Code cell 8, phylogram, fruit flies. Phylograms show branching and evolutionary distances. Let's run this cell for fruit flies. The output reveals genetic divergence, useful for studying evolutionary rates. Code cell 9, dendrogram, gene clustering. Dendrograms cluster similar items, like genes. Let's run this cell for gene expression data. This clusters genea through gene E by expression similarity, common in genomics. Choosing a tree type. Each tree type serves a purpose. Rooted trees show ancestry. Unrooted trees focus on relationships. Cladograms emphasize branching. Phylograms include distances. Dendrograms cluster hierarchically. Choose based on your study, virus evolution, species relationships, or gene expression. We've run code to visualize rooted tree, unrooted trees, cladograms, phylograms, and dendrograms, and we've seen how each type applies to real-world biological questions. Quiz. Time to test your knowledge. Let's run this cell to answer questions based on this submodule. Select the correct options as shown.